The webinar is styled towards practical applications, critical bottlenecks, and whatever you need to know if you're thinking how to set up your Deparray experiments. And I look forward to your questions. So what we'll be talking about is what is actually the role of Deparray sorting and cell selection in clinical and research molecular pathology. I will review with you critical steps in the Deparray workflow, focusing particularly on cell suspension preparation, cell staining, how to perform a successful Deparray run, and then talk about cell collection and volume reduction, molecular analysis, and how to implement Deparray in either clinical NGS workflow or how to utilize Deparray in translational research and cancer heterogeneity. So Deparray technology comes in a crossroad when you're thinking whether you want to perform bulk tumor sequencing versus pure cell population analysis. And there's no question that a vast majority of laboratories perform simply bulk tumor sequencing because it is easier, simpler, the workflows are very well established, and we know what our limits of detection are. But there is also a point where we need to discuss pure cell population analysis, and this is going to be the topic of today. The first point that I would like to emphasize, although I'm sure that everybody on this call is familiar with this, is that cancer cells do not live alone in the tumor. There's many other cells that coexist with cancer cells and end up diluting your genetic information as you're performing your sequencing. Among them are, for example, endothelial cells, which are showed here as an exuberant microvascular proliferation with red arrows, or immune cells, highlighted here with the same for CD163, showing a tumor-associated macrophages. And you can see about 30–40% of cells present in the tumor are actually tumor-associated macrophages. The quality of NGS results highly depends on tumor cell percentage. And this is just an example of one of our clinical validations when you can see a set of mutations. The font is probably too small for you to read, but it is a bunch of single nucleotide variants and small insertions and deletions at the, I would say, lower limits of detection between 10% and 2.5–3%. And you can see the detection at no dilution and then how the detection decreases as you're diluting your specimen. In addition, you can see that your ability to detect these mutations also decreases with decreasing DNA input. While your decreasing DNA input may not have much impact when your sample is not diluted, more you're diluting in, higher the impact of your decreasing DNA input. This has effect not only on tumors on variant allele frequency that you're calling, but also on the coverage, which is again highlighted here, that with no dilution, as you're decreasing, the smaller DNA input has a relatively small input. Here you see dropping out one of the known mutations, but more you're diluting the sample, more dropout you're going to have in your coverage in mutations that you're interested in. And this has been shown by many studies. It is just to reiterate that there are two factors when you're setting up your sequencing experiments: tumor cell percentage and DNA input. Even more critically, I would emphasize, is that copy number calls are highly dependent on tumor cell content. And you can see the experiment here we set up that shows you samples with various amplifications of various genes and then being sequenced and called copy number amplifications. And you can see how rapidly the number of copies called by next-generation sequencing decreases as you're diluting your specimen. While tumors that have a very high starting level of copies do relatively well even with the significant dilution 1 to 10 or 1 to 20, the genes that are amplified with a relatively lower number will be very much affected by your dilution, and you're not going to be able to call it at the threshold which is here established at 5 copy numbers. So this represents a practical problem that small and low tumor percentage biopsies are challenging for clinical next-generation sequencing, and poses the question, how do we isolate the pure cancer or stromal cells for research purposes from FFPE? FFPE material is challenging for traditional single-cell techniques. Always when we submit a grant using Deparray to NIH, there's always somebody who is smart and says, oh, why don't you just use 10x genomics? And we try to explain them that traditional single-cell techniques that are possible on a very high-quality fresh samples are simply not amenable for FFPE material. Slide scraping can improve your tumor cell percentage if the normal and tumor are clearly demarcated, but the intermixed tumor cells from the tumor stroma, as I showed you with endothelial cells and immune cells, cannot be done. In theory, laser capture microdissection can be performed, but that's a very time-consuming effort and not really feasible in clinical workflows. So the implication in practical life is that stage 4 cancers, or patients with stage 4 cancers, who are often not candidates for tumor resection, and get a small biopsy or an FNA, and that might be the only sample that's provided for the testing.